Namaste friends, welcome to Bono Biology Center for True Learning. Here I request you to subscribe our YouTube channel Bono Biology Center for more videos, updates and notifications. In this video, we learn about oogenesis in human females. What is oogenesis? Oogenesis is the process of producing the female gametes in the ovaries or formation of haploid ovum or egg in human females ovaries is called oogenesis. Oogenesis is the process leads to the formation of female gametes, that is ovum or egg, in human females. Here egg or ovum is in haploid condition. It has one set of chromosomes. Oogenesis process actually begins in the fetus prior to birth. I mean before the birth of a baby, female baby, oogenesis process started. Oogenesis occurs in the outermost layers of the ovaries. In previous videos, we came to know the structure of ovaries. In the outermost layer of the ovaries, oogenesis process happens. Firstly, primordial germ cells enters the developing ovary. Actually, primordial germ cells migrate from the wall of the yolk sac during embryonic development. During the embryonic development from the yolk sac, these primordial germ cells migrate into the developing ovary. This is the picture which shows us the primordial germ cells. This is the origin of primordial germ cells. This is the yolk sac. This is the yolk sac. These small circular structures are called primordial germ cells. Primordial germ cells differentiate into oogonia. Actually, we learn the oogenesis process from oogonia. Before that, so we need to know also the primordial germ cells from which oogonias are formed. This is the another picture which shows us oogonia in the ovaries, developing ovaries. This is one oogonia, this is another one, this is the another one. Oogenesis begins with a germ cell called oogonia. I just I have said you, oogenesis process begins with the oogonia. Oogonia is in diploid condition. We represent it as a 2n because it contains two sets of chromosomes. Couple of million gamete mother cells are formed within each fetal ovary. If we see the fetal ovary, it contains some million gamete mother cells. Gamete mother cells also called oogonia. Gamete mother cells are nothing but oogonia. The number of oogonia in each fetal ovary is couple of million. The formation of this large number of oogonia happens during the embryonic development only. During the embryonic development only. Right. One more important point. New oogonia are not formed after birth. Whatever number of oogonia present in the newborn baby ovary, that is enough. After the born of the new baby, no more oogonia not newly formed. So this is the picturization. The fetal ovary. Here germ cell cyst formation. A cyst breaks down here. Then oocyte is formed. Then enter the sexual maturation. This is the adult body. The further development will takes up to the ovulation. I mean up to the releasing of the secondary oocyte. In further videos, we will talk about the ovulation and releasing of the secondary follicle. So this is the picture presented only for our understanding. So how the oogonias are formed in the fetal ovaries. Majority of the steps in oogenesis up to production of primary oocyte occur prenatally. Prenatally means so before birth. So whatever events or whatever the steps taking place in the oogenesis happens before the birth only. Oocyte development occurs within follicle. There are some granulosa cells from which the follicle is formed. Follicle actually covers the oocyte. The oocyte development is taking place inside the follicles. Female babies born with 2 million primary oocytes approximately. The number of primary oocytes in the ovaries of newborn females is 2 million. Oogenesis and folliculogenesis occur in conjunction with each other. So oogenesis is the formation of female gametes. Folliculogenesis is the formation of follicles. Both the process happens or occur in conjunction with each other, hand in hand. Let comes to the important aspect, the phases of oogenesis. Oogenesis process occurs in 3 phases. Let us see what are they. The first phase is multiplication phase, second phase is growth phase, third phase is maturation phase. This is the diagrammatic representation of the process of oogenesis in 3 phases. The first phase is multiplication phase, second phase is growth phase, third phase is maturation phase. Let us talk about each phase in detail. The first we take multiplication phase. Multiplication phase takes place in ovaries of female fetus. When fetus is developing in the uterus of the mother, the multiplication phase happens. Firstly, oogonia undergoes mitosis to produce primary oocyte. So we can observe this happening in this picture. This is the multiplication phase. This is the oogonium, which is in the diploid condition. I mean, it has two number of chromosomes, two sets of chromosomes. I mean, 46 chromosomes. It exposes to the mitotic division and divides into two oogonia, means additional oogonia, right? Each primary oocyte is surrounded by a layer of granulosa cells called primary follicle. So after dividing of the oogonia, around the primary oocyte, so one more new layer is formed, which is called primary follicle. This primary follicle is raised from the layer of granulosa cells, group of cells called granulosa cells. Then each primary oocyte undergoes meiosis-I soon after its formation. Oogonia divides into further cells called primary oocyte. After formation of primary oocytes, they undergo meiosis-I immediately. Already we learned the cell divisions, types of cell division. One is meiosis, another one is mitosis. In case of meiosis, there are two types, meiosis-I and meiosis-II. Here, each primary oocyte exposed to the meiosis-I. Here, one important point we need to highlight and remember. In the multiplication itself, the primary oocyte doesn't complete the meiosis-I. It means the meiosis-I will be arrested at early stage of prophase-I, particularly at diplotin stage of the meiosis-I. There are different stages of meiosis-I, like prophase-I, metaphase-I, anaphase-I and telophase-I. Under prophase 1, there are the substages also. In that one of the important substages, diplostage stage, right? The meiosis 1 is arrested at prophase 1, especially at a diplotin stage of the meiosis 1. It is very important point, so we need to remember. Next, a large number of primary follicles are degenerated from birth to puberty. So as earlier, we came to know that millions of primary follicles are present in the newly born female baby. From birth to puberty, that large number will be decreased. I mean, so many primary follicles will get degenerate or disappeared or lapse. The number becomes to this small. 60 to 80,000 primary follicles are present in each ovary at puberty. If we see the number of primary follicles in the females at puberty, each ovary contains 60 to 80,000 primary follicles. So it is about the multiplication phase. What happens under multiplication phase? The oogonium undergoes mitosis and divides into two other cells. And around the primary, they are called primary oocytes. Around primary oocytes, the granulosa cells form one layer which is called primary follicle. Next, we move to second phase of the oogenesis, that is growth phase. Let us see what happens under growth phase. Growth phase occurs each month in ovary after puberty. When growth phase will start? After the reaching the puberty only. Growth phase can be seen in each month in the ovaries. Growth phase is comparatively longer than the growth phase of spermatogenesis. If we calculate the time taken by the growth phase of oogenesis is somewhat larger than the growth phase of the spermatogenesis. Next, primary oocyte become large in size. It is very important point, so which is mainly happens under the growth phase. This is the growth phase. These are the primary oocytes. In the growth phase, so we can observe the enlargement of the primary oocyte. It can become large in size. What is the reason for increasing of the size? The accumulation of nutrients and other materials synthesized. There are some nutrients and the materials are synthesized and accumulated in the cytoplasm. Due to this reason, the primary oocyte so become large in its size. Here, yolk is accumulated, so which is also called vitellogenesis. Accumulation of yolk is called vitellogenesis. Here, large amount of fats and proteins concentrated towards vegetative pole. Here, primary oocyte has two poles. I mean, the primary oocyte cell has polarity. One is animal pole, another one is vegetative pole. This is the vegetative pole, this is the animal. The vegetative pole, towards the vegetative pole, the large amount of fats and proteins are concentrated. Next, come to the animal pole. The animal pole also with cytoplasm with egg pronucleus. Then DNA synthesis is also the formed. Then other substances like so DNA, RNA, then tRNA and some enzymes also present in this. The third phase is a maturation phase. Here, this much of thing is a maturation phase. Let us see what happens under maturation phase. Here, every primary oocyte experiences two maturation division. This is the primary oocyte. After completion of the growth phase, each primary oocyte enters the maturation phase. When it enters the maturation phase, it exposes or it experiences two types of divisions. Primary oocyte firstly divides into two uneven cells. This is the primary oocyte. Firstly, it divides into two unequal cells. This is the large cell. This is the small size. This large cell is called secondary oocyte. This small cell is called first polar body. So if we see the condition of the secondary oocyte is a haploid condition. I mean it has only one set of chromosomes. When primary oocyte, which is in diploid condition, divides into two other cells, the two sets of chromosomes are separated and enters into two unequal cells. One set enters the secondary oocyte, another set of chromosomes enters the first polar body. But the nutrient-rich cytoplasm is retained in the secondary oocyte. The less amount of cytoplasm is present in polar body, but the large amount of cytoplasm, which is rich in having the nutrient, is retained by the secondary oocyte only. Polar body has a little amount of cytoplasm with one set of chromosomes. Compared to the secondary oocyte, polar body has a very small amount of cytoplasm. But in case of having the chromosomes, as secondary oocyte contains one set of chromosomes, the polar body also contains one set of chromosomes. Then secondary oocyte begins meiosis-II, but arrested at metaphase-II up to the fertilization. Here, secondary oocyte after its formation immediately start the meiosis-II, but this division do not continue to the end, but in middle it will be arrested. The phase at which second meiosis-II is arrested is metaphase-II. When the meiosis-II will complete at the fusion of both secondary oocyte and sperm, I mean male and female gametes, the meiosis-II will be so open and proceed further to complete it. At fertilization, secondary oocyte finishes meiosis-II and form mature ovum along with two polar bodies. So we can observe in this picture, this is the secondary oocyte, so exposed to the meiosis-II. At the end of at the time of the fertilization, the mature ovum is formed and the secondary polar body is also formed. So here, little information is additionally provided about the polar bodies. How many polar bodies are formed during the oogenesis process? So there may be 2 to 3 polar bodies are formed. The first polar body is formed during the meiosis-I. So here the primary oocyte exposed to the meiosis-I and divided into two unequal cells. One is secondary oocyte, another one is the polar body. This is the first polar body. The first polar body is formed during the meiosis-I. Then come to the second and third polar bodies. They are formed during the meiosis-II. When secondary oocyte undergoes meiosis-II, two unequal cells are formed. One is matured ovum, another one is the polar body. This is the second polar body. Sometimes the first polar body will divide further and form two new polar bodies. They are second polar bodies. Here, polar bodies do not contribute genetic mixture to the zygote or fetus. Polar bodies contain one set of chromosomes as secondary oocyte contains. Even though they contain the chromosomes or genetic material, they do not share it to either to the zygote or fetus. Finally, we brief the oogenesis process. The oogenesis process starts with the oogonium. So oogonium contains two sets of chromosomes, I mean 46 chromosomes. Firstly, oogonium undergoes mitotic division and forms additional oogonia or primary oocytes. They are also in diploid condition. This multiplication takes place in ovaries of female fetus, I mean in unborn female body. Then the primary oocyte enters the second phase, that is growth phase. During the growth phase, the primary oocyte increases in its size. The reason is the accumulation of nutrients and other substances. It occurs each month in ovary of female after puberty. Multiplication phase happens before the birth. The growth phase occurs after the puberty. Then after vitellogenesis, after completion of the growth, the primary oocyte enters the maturation phase. Here, firstly it exposed to the meiosis-I and it divides into two unequal cells. One is secondary oocyte, the another one is the first polar body. The secondary oocyte contains large amount of the cytoplasm with one set of chromosomes. The first polar body contains a little amount of cytoplasm with one set of chromosomes. Then The secondary oocyte that begins the meiosis-II, but it is arrested at the metaphase-II. At the time of fertilization, the secondary oocyte completes the remaining meiosis-II and forms the mature ovum and another polar body. There are 2 to 3 polar bodies are formed in the oogenesis process. The first polar body is formed during the meiosis-I. The second and third polar bodies are formed during the meiosis-II. The polar bodies do not share any kind of genetic information to the zygote or fetus. Thank you for watching this video.